just open this up right here. For all of you that have read the program, and know I'm obviously not Robert Schultheis. I'm Dr. Frederick Gertz. I work with Robert, and so I've come to present the talk. He apologizes for not being able to be here, but he sent me in his stead. So, so like I said, I'm Frederick Gertz, and we're going to talk today about mitigating risk associated to auto-injector applications. So it's a bit different than the fundamental research that most people have talked about today, but it is concerned with the novel drug delivery system, which is auto-injector devices, and sort of problems associated with them, and some of the things we found out at ZebraSci and Mantis Vision Systems concerning how to mitigate the risks of auto-injector devices performing incorrectly. So get right into it. A quick overview, I'm going to talk a little bit about what the risks are prevalent in auto-injector devices. And then once you've identified these risks, what are the most important variables to explore to determine which aspects will give you the best device, the device that will act in a way according to your design specification. So it's a real industrial problem that comes after you've invented some sort of novel drug delivery system. There's a lot of questions that come into quality control for people using them. And then we'll talk these last two lines, which is what is the effect of lubrication and what aspects of lubrication are the most important. Right now they don't make a lot of sense, but we'll come around to why it's important that these devices are lubricated in a manner that's appropriate, and not just appropriate in general, but appropriate for the individual device. So a little bit about the auto-injector market. Auto-injectors are a drug delivery mechanism. Very simply, they're essentially syringes that a person doesn't have to press on. So it's a syringe usually with a plunger controlled by a spring, though there are some applications where they use electric motors, or in some cases we've even seen people creating a gas using electrolysis to push the plunger. And in and of itself, it's about over $500 million market. For those of you not familiar with it, you've probably, the auto-injector you're most familiar with are those associated with what are called EpiPens, so delivery of epinephrine for people that have gone into anaphylactic shock. The auto-injector itself is a form of risk mitigation. It removes the problems associated with somebody overdosing or underdosing. It also removes a lot of risk associated with cross-contamination. Currently, if you use a syringe, you pull from the same vial with several syringes. One of the syringes was contaminated. It may contaminate all subsequent syringes. As well as you have some problems with usually you overfill a syringe and then you eject some of it out to remove air bubbles. So you end up with the possibility of incorrect dosing. You end up with a lot of wasted drug product. And it's, of course, something that's usually injections are usually supervised by a doctor, so they're not very convenient for a patient, and especially a patient that needs a lot of injections. So within this, there's a variety of risks associated with the production of auto-injectors. And some of these make sense, right? It's an unsupervised injection. You're not giving an injection to someone who's trained to give injections. There's no supervision of a doctor. They don't know how much of the drug may be important, how much over and underdosing could affect them. They're not aware of timing of dosage. So a lot of these are concerns that have to deal with just patient treatment. But there's also risk associated with any sort of manufactured project that are inherent in manufacturing. So typical risks that you associate with the syringe and a cartridge, and even what we use normally for syringes, are things like drug stability. Will the drug remain stable when you put it in these devices? Is the fill volume correct? Meaning, since these are parental syringes and they contain drug product, do they have the correct dose? What is the needle quality? Is the needle broken? Will the needle shield extend? A lot of these problems are classical problems within quality control and can largely be handled by large-scale producers and manufacturers of auto-injectors, such as SHL. These groups have a lot of control, but the most important control aspect for an auto-injector is what we call injection time, meaning how long does it take to inject the drug product? Is that injection time consistent? And if it is consistent, how often do these fail? So the two big problems you run into with the production of an auto-injector is either long injections or what we call catastrophic failure, stalls, where the entire product is not injected. Long injections, you have the problem of some people will pull the needle out before it's done injecting. And of course, with stalls, people are not receiving the amount of drug that they thought they were receiving. Failures of these devices have resulted in several $100 million in recalls, especially for devices such as Enbrel, which I've shown, and Simponi, which is shown below it. But even devices that haven't failed yet, there's a lot of devices coming to the market, especially for arthritis treatment, which we just heard about, typically rheumatoid arthritis, where people are going to need a lot of injections over time, unsupervised, without the ability for to have that knowledge that a doctor would have. So you're seeing a problem that causes hundreds of millions of dollars and has delayed, to this point, hundreds of millions of dollars of more drug product being delivered. So if you look at rheumatoid arthritis treatments that are coming out now, a lot of the products that are hoping to have auto-injector delivery in the future or the near future are seeing problems with these devices, and they're having to delay the release. So let's get into it. What factor most affects the failure or passing of one of these auto-injector devices? And I have here sort of a simple plunger, and you can see that at some point you're delivering product. It's a hydrodynamic problem. I originally studied physics, and it seems like you boil it down, it's a simple hydrodynamic problem. You can write out this Poisson equation, and you can see that if you account for the flow rate, the viscosity, which is controlled by the drug product, the length of the needle, which is controlled by your supplier, say BD, or the internal diameter of the needle, and the pressure drop, you can write out this simple Poisson equation. And I don't want to bore anybody with too much math, but what's very typical here is what people get hung up on is you see that the radius, the internal diameter of this needle, has sort of this largest effect, right? It's to the power of 4. Along with that, there's just some common sense effects that you think. The plunger, for example, comes in direct contact with the barrel wall, and you suspect that if there's a failure, a stall, that it has something to do with the plunger and its friction as related to the barrel wall, which isn't an incorrect assumption. There's also, you want to explore the power of the power pack, which is the spring pushing it. All of these dimensions make sense, and you would think that throughout a study, especially a lot of root cause investigations like we've conducted with thousands of devices, you'd see a variety of problems that are usually resulted in some sort of quality control, which is true. What we've learned is the failure of these devices tends to be a quality control issue, which we'll see, and it's a quality control issue that's sort of for an unrelated topic to what people inherently think it's going to be. Most people think it's a problem with the plunger, it's a problem with the needle, and so we've explored those. So the first thing we explore is we've picked a group of topics. Let's see, and so let's say for our standard root cause investigation, we may study barrel dimensions, its eccentricity of the barrel, is the barrel round, as well as the plunger dimensions, the plunger composition. We'll study whether the power packs are strong enough using an instrom and a material tester. We'll study needle diameter. The two that are usually sort of green-lighted, the things that most companies think will be the problem are the plunger, which has sort of the least amount of control. So here I've got plunger dimensions, and we actually have automated, since we are inherently an automation company at ZebraSci dealing with data in large amounts of it, we actually automated the study of plungers and put over 1,000 plungers through what we call the plunger dimensioner, for lack of a better creative name. In the plunger dimensioner, if you look here, these green lines, it shows we measure 11 different dimensions along the plunger. We do this automatically. We do this for 8 different rotations of the plunger. We can do this very quickly. It's all done using computer vision. And for an example here, you can see that I picked L parameter, which is the trim edge. The trim edge is the widest point of any plunger, and also the most difficult to measure. And of this point, we then looked at the distance of every plunger measured, its trim edge from its average trim edge. Now, what we would hope to see is almost all of them stacked right along the middle of this Cartesian graph, and what you'd see is a scatter point that's very, very tightly grouped together. What we actually see is there's a ton of spread within these groups of tens of microns, and it's not a lot of control over the plunger. Now, that, of course, seems to be a problem. And then I even put a 1 standard deviation circle showing you, okay, this is where you would hope that at least all of your plunger dimensions would fall within 1 standard deviation. They don't even come close. You have something about a little over 40% fall outside of this when we do large studies. So the plunger is not controlled, and of course you think that's going to be the problem. However, when we look at correlation of plunger dimensions, even the ones that fall well outside this, we find very little correlation to either glide force, which is the amount of force it takes to move the plunger, or injection time. The next big one that you might look at is needle ID. So this is a microscope image of the backside of a needle. And you can see that the needle ID here is 233 microns. These are 270 thin wall. These are 27 gauge thin wall needles, so they should have an internal diameter of 270 microns. So I have kind of a bad example here. But when we look at thousands of examples, we see exactly what we would hope and expect to see. We see a normal distribution. It's centered around 270 microns. And in fact, there's a slight increase towards things with a wider needle, which would result, hopefully, in lower injection times. The moral of this story is we get to look at a lot of different dimensions. And when looking at these dimensions, we do see variation. We sometimes see unexpected variation in the example of plunger dimensions. And these variations don't seem to actually correlate all the time to device performance. Of course, there are some devices that have probably performed poorly because the needle diameter is too low. Therefore, the force is too high. Some have performed poorly because there was an error in the spring manufacturing. Some have performed poorly because the plunger maybe is incorrect. But we never see a consistent story. And what we do see is a consistent problem in these devices where we see long injection times. So in our search, and what this relates to is something that started about 5 years ago, over 5 years ago by the president, Robert Schultheis, of our company, was to find a mitigating factor that causes these long injection times. And what's interesting is he discovered, and what we've really kind of proven, and we'll show sort of some data on, is that there's a lot of variation in these devices, but the one thing that most companies don't control is the lubrication of the syringe. The syringe is actually lubricated with silicone oil. It's a biocompatible oil that's used all the time in pharmaceutical industry. And that oil is bought. You buy from a supplier, say OMPI, BD, Gersheimer. It is sprayed on the inside, and large manufacturers buy these already siliconized syringes, and then they fill them. Nobody ever checks the quality of the siliconization. Now, to an extent, it's a hard problem. Silicone oil is clear. Some of these films of silicone oil are as thin as 60 nanometers. It's like any sort of spin coating process. It's a dynamic liquid. It's difficult to measure. There are only a handful of systems on the market that can do it, our system at ZebraSci being one of them. It's interesting also that most people do not realize that when you add a drug product to your syringe, that the silicone oil goes from a thin film to beading up. And now, because it's immiscible in water, it's de-wetted off the surface. These beads can move. When you ship a syringe that has been lubricated, the lubrication changes. So we have some device here. I'll show you. This is a close-up of a lubricated syringe. And this is typical of what we see in syringes. So we see a raw image. It's a black and white image. And underneath, you can see we've used an algorithm to detect where the silicone oil is. It's the white area. We're then able to determine the percent coverage. What do we see? About halfway down the syringe, there's no more siliconization. Well, that's almost the worst place to be, because where's the spring weakest on an auto-injector device? At the end of its injection time. It's the place you probably need the most lubrication. This is a typical pattern that we see, heavy towards what we call the flange end, none or light towards the needle end. So we spent a lot of time not only building devices that can inspect this, but also giving us an ability to see that how prevalent, how important is siliconization in the mitigation of auto-injector risk, meaning bad injection times or stalls. We've been able to form these heat maps, and you can see this is a heat map from something that's been shipped. You can see there's a slight change of the heat map over several weeks. So each one of these represents 1 week at a time in an incubator. And you can see that if you leave these syringes out, the silicone oil indeed does move. And it's hard to see in these images, I'm sorry, because they're projected. But you can see in these other images that you can actually see the silicone oil droplets. They're moving. And so we have this ability to not only take these images, we have an algorithm that detects silicone oil, and then using that detection of silicone oil, we're able to tell people not just where the silicone is, but we can do studies to tell them what sort of distribution is important. So how important is it? Well, here's an example of glide force, where we take an Instrom or any other materials tester, and we actually trying to detect how much friction force, how much force it takes on the plunger to move it. And we've done a correlation with silicone coverage. So the black line represents glide force, which is the amount of force needed to move the plunger. And the red line represents mean silicone coverage, the average of a radial slice. And as you can see, makes sense. If you've put something on the surface of a syringe that was to reduce friction, and there's less of it, you induce more friction, and therefore, you have a higher glide force. And we get a pretty good, almost one-to-one correlation. It's quite apparent that the glide force and the coverage are negatively correlated. You need good coverage to get good glide forces, and you need good glide forces to get good injection times. So lubrication appears to have a very large effect on the glide force. It sort of makes sense once it's apparent. It sort of makes sense that it can be inspected. But there's very little control of this one aspect of silicone coverage. The reason being, it's always thought that it's one of the other classical quality control problems. So now the question is, how important is lubrication? I think everyone believes me that if you have something to reduce friction and there's not enough of it, then you have too much friction, there may be a problem. So on the left, I have a graph here, and this is a correlation of coverage to glide force performance. Basically, we've taken glide forces and we've done a scatter plot, and it's very obvious that these are correlated. Not damning evidence in and of itself, though. You would say, okay, we buy into the idea that tribological science has taught us that friction is a problem in these devices, and that, of course, if we don't have enough of this. But how well does it actually relate to device performance? So on the, I guess, your left, my right when I look at it, we say, looking at what we consider good coverage, how well can we predict the performance of an auto-injector? 2 minutes? Okay. In the case here, you can see that with 99% effectiveness, we can tell you whether an auto-injector will pass or fail in some cases based on how much silicone coverage there is. In this case here, we tested several hundred devices, but only one device was there a fail that we incorrectly tested. That's a huge number. 99% of products that people are making millions of, we can tell you just by looking at one variable that you can inspect industrially whether or not that device will work. This is a good example of good and bad silicone coverage. I think we've all believed in it. You can see a bad distribution here next to a good distribution and their related injection time. I'll kind of go through it. And this is to say that the other important thing that industry usually asks us is, well, how do we determine the amount of coverage? And what we try and harp to our clients is the amount of coverage is not nearly as important as the amount of silicone oil is not as important as the distribution, the coverage. In this case, we're able to see that if you put in 0.8 mgs or milligrams of silicone oil, you actually don't get as good a glide force as a well-distributed silicone oil done with a diving nozzle, where we used almost half, 0.5 milligrams of oil. So using almost half the amount of oil, which decreases the amount of protein aggregation, the potential for leachables, you were able to actually get a better glide force using less silicone oil by using a diving nozzle, which gave you a better distribution. For these devices, we're even able to use advanced statistical analysis, in this case a support vector machine. And by using this, we're able to not only identify for each product, we're able to identify for each product an area that has to be inspected. So there are the old common sense areas that we used to use as ZebraSci inspection, where we took good and bad lots, we looked at their distribution, and we said these are the areas that are most important. But now using a support vector machine and other regression techniques, we're able to actually tell you that there are thinner slices to inspect, as well as points in the middle that we hadn't obviously thought to inspect. By using these tools, not only can I tell you for your device how much your silicone coverage should be, but I can tell you where it should be. Instead of just telling you you need the mean coverage of this, I can tell you need a mean coverage toward the needle end and maybe this much towards the flange end. This is a powerful tool for people to mitigate the risk in their auto-injector devices. So with these new distribution curves that we're able to provide to clients, or help them provide it themselves using our equipment, you're able to mitigate a huge amount of risk by studying the distribution pattern of your good and bad lots, and by replicating the good lot distribution and getting much higher failure rates for not a lot more money. So yeah, in conclusion, auto-injectors represent an increasingly important delivery system in the market. Physical dimensions and other factors play an important part in the performance, but even if you have a lot of variation in other factors and poor quality control, if you have enough silicone oil distribution, then you will get a good performing device. So it's not necessarily about the amount of silicone oil. It's about the coverage and how well you covered the silicone.
think you are feeling very hungry, or still, if you have at all any.